This is a production of Cornell University.
So, as many of y'all know, our lab works with soil microarthropods, which are primarily columbolan mites and are tiny animals in the soil that we really care about because of their effects on the decomposition of organic matter as well as nutrient mineralization. And one of the primary ways that microarthropods are affecting these processes are through their interactions with microbes in the soil, especially through grazing and feeding on these microbes. So soil microbes, a lot of you work with, but for the sake of today's talk, I'll be focusing on the saprotrophic microbes that are important for nutrient decomposition. And as we all know, they're not just found randomly in the soil. They, like us, aggregate around the resources that they need to live, for example, an alfalfa leaf. However, due to their small size, they're not just able to take a bite out of that alfalfa leaf. They instead excrete extracellular enzymes into the soil environment, which then helps to release the target nutrient that enzyme was designed to break down into the soil environment, at which point the microbes that did the work can either take it up or it can be absorbed by plant roots in the soil. At this point, we're still not really sure how microarthropods are affecting this process. So we wanted to design an experiment where we could look at how microarthropods are impacting soil fertility processes and see if we should be considering soil microarthropods when making soil fertility management decisions, like a farmer picking which fertilizer he's going to use. To do this, we set up a greenhouse experiment with 3 different microarthropod community abundance levels. We had a no community control treatment, and then a low abundance and a high abundance of the microarthropod communities. We then chose some fertilizers that are commonly used by organic farmers in our area. We had a no fertilizer control and then used Crayer's poultry litter compost, alfalfa green manure, and Chilean nitrate, which were all chosen due to their different nutrient compositions and carbon to nitrogen ratios, which affects their plant availability. To set this up, we defoliated a local soil as well as potting mix, and then extracted native microarthropod communities from a local pasture, which would be comparable to what you would see in an agricultural field, and extracted them into those 3 bulk soil bins using Berlese funnels. We then applied our fertilizers at a standardized rate of nitrogen to keep things equal, as well as ground them up so it was all homogeneous, and then sowed our oats into the pots at an equal rate. The oats were then allowed to grow for a little over 8 weeks, and at each week we would go in and do a check where we measured the oat growth stages, the number of seeds that had germinated, the height of the tallest plants, and just overall plant health observations. Then at 2 different time points, we harvested half of the pots. Our first harvest was timed at around the boot stage to mimic a forage harvest, and then the second harvest was timed at around the soft dough stage to mimic a grain harvest for farmers. And at each of these harvests, we measured our oat shoot, root, and seed weights, as well as weed weights by clipping, drying, and weighing that biomass. We measured soil microbial biomass using a total organic carbon and nitrogen analyzer, and then microbial enzymes using multi-well colorimetric and fluorescent assays. And lastly, we wanted to check our microarthropod community treatment abundances using Berlese funnels, which is what I'll highlight first, because while this is a treatment, microarthropods are living organisms that the communities are changing over time, so we wanted to check and make sure that our treatments were true. At the initial setup, we had 3 distinct abundance levels for our microarthropod community treatments like we were aiming for, and we also had 3 distinct different taxon richness levels. So as the abundance of microarthropods increased, so did the number of different taxa we were finding. However, by that first harvest, our abundance of the microarthropod communities had an interesting interaction effect, which, as you can see here in our no community pots, we basically still had 0, luckily. But then within the no community treatment, the green manure had really shot up our microarthropod abundances, significantly more than all the other fertilizers, and we saw a similar trend starting to occur within our high microarthropod community treatments, but it just wasn't to as high of an extent. Then continuing on to at the end of the experiment, that last harvest, instead of that interaction effect for our microarthropod abundances, we had distinct fauna and fertilizer treatment effects. And here we did have no difference between our low microarthropod and high microarthropod community, but they were both greater than our no microarthropod community treatment. And within the fertilizer, the green manure was just far above in microarthropod abundance as all the other fertilizers. So now to dig into that weekly check data, here I'm showing you the growth stages of the oats, or how far along they've developed over time. And here you'll see that the no fertilizer treatment is represented by gray, compost is represented by yellow, green manure is represented by green, and Chilean nitrate is represented by red. But the main thing I want to point out here is how the green manure, especially near these last few weeks, is really lagging behind developmentally as far as growth stages. But interestingly, when we flip over to plant height, it's the green manure that's significantly taller than the other fertilizer treatments, especially the no fertilizer treatment here, which was quite short. Continuing to focus on that oat biomass, by the first harvest, you can see in this graph on the left that the compost and Chilean nitrate treatments had significantly more shoot biomass production compared to the no fertilizer and green manure treatments, likely because earlier on in the growing stages, these two fertilizer sources were more plant available and gave them more of an edge. Then focusing on this graph on the right, you can see the root biomass, and as the fertilizer source becomes more plant available, we see that our root weight is decreasing, which indicates that when there's more nutrients available in the soil, they don't need to scavenge as much and put on as much biomass. By the second harvest, we see similar trends for both the shoot and root biomass, just exacerbated a little further, with the shoot biomass of the Chilean nitrate being significantly greater than all other fertilizer treatments, and the root biomass in the no fertilizer treatment being leagues above the rest of the other fertilizer treatments. Also at that second harvest, we had seed production, and I'm showing you seed biomass on the left and seed count on the right, which look basically identical because there was such a high correlation between these two measures. You can see that the compost and Chilean nitrate treatments both produced more seeds than the green manure and fertilizer treatments. However, if you'll recall back to that growth stage chart I first showed you, the green manure was developmentally behind the compost and Chilean nitrate, and I think had it been given more time to develop, it would have had more comparable seed production to those two fertilizer treatments, but we just had to pick which timing was best for the majority of pots developmentally. I did want to jump back to the first harvest really quick to talk about an interesting finding with our weed biomass. For context, at around the second week here, we thinned out all of the pots and we removed any weed biomass. So what you're seeing here with the weed biomass of that first harvest is really more of an indication of if weeds had germinated in the pot or not. And as you can see in this graph, whenever we had our high microarthropod community abundances, they had produced more weed biomass or stimulated weed germination more so than the no and low community treatments. And we can attribute this to those microarthropods grazing on those fungal seed coats, which will stimulate weed germination. Digging into the soil a little bit deeper, here I'm showing you a busy chart about the microbial enzymes we measured at the first harvest. You'll notice we measured enzymes for chitin, cellulose, amino acid, phosphorus, and lignin breakdown in the soil, all of which had interesting interaction effects at that first harvest. But there are some overarching trends, like within the no microarthropod community treatment, you see that the no fertilizer generally produced more enzymes than the other fertilizer treatments. However, whenever there was a lower abundance of microarthropods present, some of these effects tended to disappear, though when they increased, there was an interesting flip where it was a specific fertilizer treatment for each enzyme that was being stimulated by that high microarthropod community compared to the no fertilizer. But for the sake of time today, I really want to dig into this one enzyme, NAG. And we really care about NAG because it's important for breaking down chitin, which is an important component of fungal cell walls. If you look to the far left here, you'll notice in that no microarthropod community treatment, as the fertilizer source becomes more plant available, there was less enzyme in the soil to break down NAG, which makes sense because the plants did not need it broken down and the microbes didn't either. However, when we flip over into the low microarthropod and high microarthropod community treatments, it's the green manure that was above a lot of the production within the different fertilizer treatments. If you recall back to that microarthropod check data at this first harvest, there was greater microarthropod abundances in both the green manure treatments for low and high community, which suggests that the microarthropod presence are having a stimulatory effect on the NAG production of the microbes. Quickly wanted to touch on the microbes at this microbial enzymes at the second harvest. Here you'll notice that there's not as many interesting interaction effects between the microarthropods and fertilizer treatments, but what is there is very continuous as to what we saw at the first harvest. But we didn't want to just look at these plant metrics. We wanted to explore the relationships between the soil and plant metrics we were able to observe. So here I'm showing you at that first harvest a relationship between oat biomass and NAG. This is the shoot weight here on the first axis and NAG on the x-axis. So as NAG is increasing, the shoot weight is decreasing. But interestingly, when we look at the relationship with the root weight, which was also significant, the increase of NAG would cause an increase in the root weight, suggesting that the presence and breakdown of nutrients in the soil are in fact affecting how the oat biomass is being partitioned in the plant. At the second harvest, there was still that trend between the shoot weight and NAG. However, by this time point, the root biomass relationship had disappeared and was no longer significant, which is likely due to the need for other nutrients aside from nitrogen within the pots that would affect the root biomass production. Also at that second harvest, as I mentioned, we had seed production. I'm showing you seed count and seed weight again, and both were of course negative relationships with NAG. So as NAG was increasing, the seed production was decreasing, again reconfirming our idea that the NAG production of soil fertility was affecting how the oats were growing. So just to quickly wrap up, we see how fertilizers in the study are affecting our microarthropod and microbial communities in really diverse and interconnected ways that aren't clear or clean cut for making management decisions. Because it's not just affecting these communities, but also the activity of these communities, which ultimately affects the soil environment and how the plants are going to grow. So to end, I'd just like to thank my funding sources, as well as the Wickings Lab and all the techs that helped make this possible.
This has been a production of Cornell University, on the web at cornell.edu.